So picking up where we left off, there are 4 coronaviruses that cause the common cold. There are then 3 that have emerged more recently. Coronaviruses are enveloped and they have these large spikes on the surface. The basic structure of them looks like this. The genome, as we mentioned last time, is either DNA or RNA for viruses. Coronaviruses have a single-stranded RNA genome. And so there's kind of a cartoon where you can see the single-stranded RNA genome surrounded by a capsid, and then that is all encompassed or contained in this envelope, this membrane, and then there are those spikes that project out from that. So that's a bit of a background. Let's now move into basic symptoms of the coronaviruses that you have likely been infected with. So the incubation period is about 3 days. It's relatively short. And the thing here with these other 4 coronaviruses is largely the infection and the disease is upper respiratory tract. So for the most part, the symptoms that you get are sort of nasal discharge, that snot and fatigue. It's not lower respiratory like we're seeing with SARS. Lasts about a week. Again, cough, fever, sore throat aren't as evident with the common cold coronaviruses. Epidemics of this, so outbreaks are typically winter, early spring, every few years. So this has been something that's been talked about quite a bit recently of, can we expect this new virus to die down after the spring? Well, the reality is we just don't know. Every virus has unique properties, and so we just don't know, and we'll see. Most of us, again, have been infected with this, and reinfections are common because generally the immune response to these common cold coronaviruses is not all that strong. So reinfection is possible. Again, this brings up lots of questions with the new coronavirus. How's the immune response going to be? Can we get reinfected? Once we're infected once, are we susceptible again? Again, the reality is we just don't know. This is a brand new virus that the human population has not seen before. So again, these are the facts that we know from the coronaviruses that cause more like common cold symptoms. We just don't know how this new SARS-CoV-2 is going to behave. So that kind of brings us to the newly emerging ones. And again, that began with SARS back in 2003. So SARS, severe acute respiratory syndrome, again, emerged in 2002 under sort of similar circumstances in China. 2003, it had spread worldwide. The final tally, though, was 8,460 people, of which about 10% died, so relatively high mortality rate. And again, much like we're seeing with COVID-19, with the new SARS-CoV-2, is that the mortality rate is higher in the elderly. And so for SARS, there was about a 50% mortality rate in those that were above 65 years old. In the end, it had spread to 32 countries. There were only 8 cases of the original SARS here in the US, and they were all cases of folks that had traveled abroad. There was no evidence of community spread, obviously, unlike now with SARS-CoV-2, where there is a tremendous amount of community spread at this point. So original SARS, about 8,000 cases, about 800 deaths. Only 8 in the US, no evidence of community spread. The symptoms of that largely, again, upper respiratory symptoms, but the big difference was lower respiratory, getting into the lungs and causing pneumonia. And you can see the timeline here in the figure. Up at the top in blue are the particular symptoms that are most common. And then at the bottom, you can see how the virus is shed from the body. And so you can see nose and throat is evident, but also in the stool. And you can see for quite a while. So the incubation period of SARS, the original SARS, was about 10 days. High fever was evident, again, a little bit different than the common cold coronavirus, but dry cough, fatigue, high fever, again, similar symptoms to what we're seeing now. One of the unique symptoms, though, was that with the original SARS, about half of the patients had some kind of diarrhea. That's not really being seen with our current COVID-19 disease as a result of SARS-CoV-2. Symptoms peaked in about a week. And again, most did develop pneumonia. And that was the really key kind of lower respiratory infection symptoms that would progress potentially to some really significant respiratory symptoms. Again, where 10 to 20% would require respirator. And then again, overall mortality rate was about 10% for SARS. The interesting thing, again, as you can see from the chart in red, this virus was shed for about a month. So nose and throat up to or exceeding about 30 days. And because of the diarrhea and the infection there, also present in the stool for about 30 days. Again, it's not something that we're seeing really with this new SARS-CoV-2, but something certainly to keep an eye on. All right, so here's one of the questions that's pretty significant. Where did SARS come from? And this kind of sheds light onto what's happening today as well. So the original SARS was essentially determined or eventually determined to come from particular species of bats, and so specifically Chinese horseshoe bats, and it's believed that that's kind of the reservoir for the virus. That's where it hangs out. That's kind of the natural host for it. But there may be other animals involved in the transmission, so sort of like an intermediate between the bats and humans. And for SARS, the original SARS, it was determined, or at least there's a theory that it's from civet cats. And so you can Google that and see pictures of them, but these are also in the Chinese markets and they're also eaten. And so that can be, a route of transmission. And so the original SARS, again, largely determined that the reservoir for it is these particular bats, but there might be an intermediate animal like civet cats that's transferring it or transmitting it to humans. So why the difference in disease? Why are there 4 particular strains of coronavirus that cause common cold symptoms, largely upper respiratory tract? And then we have new emerging SARS-CoV-1, MERS, SARS-CoV-2 that cause significant lower respiratory infections, damage to the lungs, requiring respirators, ventilators, et cetera. Well, one of the things, again, we talked about last time, that viruses bind to their cells through that spike protein. And on the surface of the cell is a particular receptor, a particular protein typically, that spike protein binds to. Well, the interesting thing is that these particular coronaviruses use slightly different receptors. And depending on the particular cells that those receptors are located on, that can change, obviously, which cells the virus infects, and then the disease that results from that infection. So we know that one or more of the common coronaviruses that cause common cold use a protein called APN. MERS binds to and uses a protein called DPP4. And SARS-CoV-1 as well as SARS-CoV-2 use angiotensin converting enzyme 2 or ACE2. So SARS-CoV-1 and SARS-CoV-2, again, they bind to a different cell receptor, a different receptor on the host cell than does the common cold coronaviruses. And so this may be a reason, again, why we see a different disease resulting from those different infections. Okay, so that's kind of the history lesson, the background. So again, coronaviruses have been around for a long time, circulating in humans. There are 4 that causes common cold or that causes a common cold-like symptoms. But again, there are 3 that have recently emerged. SARS, the original SARS in 2002, 2003, MERS a little bit more recently, 2012, 13. And then we see now, obviously, SARS-CoV-2 emerging and causing disease here late 2019, 2020. So let's kind of focus on that. And I'm going to be showing you quite a bit of data. So as non-science majors, I don't want you to freak out. But I think it's important to show you data so that you can be aware of what the science says. You see lots of opinions out there. Everybody has one. But really the value, for me as a scientist, the value in any opinion is on the evidence that it's based upon. And so if it's not based upon evidence, then it's just that, somebody's opinion, and you should kind of take it with that knowledge or with a grain of salt. So I'm going to be showing you quite a bit of data. I'll try to guide you through the data. Again, don't panic when I put up graphs and figures and things from papers, but doing the best job I can. There's papers rolling out every day, trying to keep up, trying to show you what's the latest, what does science say, what do we know about this particular virus, this particular disease. So here we go. Let's get some terminology out of the way. So COVID-19 is the disease. So to be clear, that's the disease that results from an infection by the SARS-CoV-2 virus. So to be clear, SARS-CoV-2 is the virus, COVID-19 is the disease that may result from the infection of that virus. So just getting that terminology out of the way. First case is, as you know, November 2019 in Wuhan province of China. More terminology to get you up to speed. An epidemic is a single outbreak in a particular location of some disease. When that epidemic then begins to spread and you see epidemics worldwide, then it becomes a pandemic. So that's the difference between an epidemic, kind of a single episode, and a pandemic is when it goes worldwide. That's obviously the situation that we're in now. We are in the midst of a worldwide pandemic as a result of this particular virus. Something else that's going to be important for us to talk about. This is a virus, I used the word emerging. So this is a virus that has come from a different animal source, a different species, and has now infected humans. And so we call that process zoonosis. So it's the transmission of a virus or something from one particular animal species to another animal species. So we can refer to this virus as zoonotic, and the process is zoonosis. And so it's important that, again, we just get some terminology. Words matter. Absolutely, in science, words matter. And so it's important for us to understand the terminology that we're going to be using. So epidemic, pandemic, zoonosis. Okay, so what are the symptoms of this thing? Again, you have heard these, but just to make sure that we're all clear, fever, dry cough, and fatigue are the three. Those are the big symptoms that again, they're the symptoms of lots of things, but a fever, a dry cough, and a fatigue, those three things are really the symptoms, at least initially, of COVID-19. And then it can progress from there. But it's important, again, that we recognize that. And again, compared to the original SARS, where about 50% of the patients had some sort of diarrhea or GI issues, that doesn't seem to be the case with COVID-19. Some numbers that I saw out of China were maybe 3%. So it seems as though diarrhea is not a common symptom that's caused by COVID-2. So here we go. What's our current status? Where are we? So as of this morning, so just before we met, we are almost at 500,000 cases worldwide. So almost at 500,000 cases. There have been 22,000 deaths. And I know that these are just numbers. I hate to reduce human life to just a number, but this is kind of where we are. So about 500,000 cases, 22,000 deaths. And we're at or exceeded 70,000 cases here in the U.S. Currently, the U.S. Is right around number 3, but that's probably going to change very quickly. Right now, China's number 1 in terms of number of cases, Italy number 2, and the U.S. number 3. But again, that's going to change here in the next couple of days. Mortality rate is all over the place. It's really highly variable. You've probably read a lot about this. In Italy, it's about 10%. Which is absolutely terrifying. So they have on the order of, about, again, 70,000 plus cases, and they have about 7,000 deaths. And so the mortality rate there is pretty high. Here in the US, we're only at about 1.5%. So why that's the case, it's just not really known, but it should not be obviously taken lightly. Again, Italy and the places where the mortality rate is high, there are cutting edge hospitals trying to take care of these patients. They're just completely overwhelmed. So it's a little bit too early to try to figure out why the difference in mortality rates. But the reality is that from country to country to country, the mortality rates are quite variable. Okay. So what's the origin? Where did this thing come from? I use the word emerging. Where did it emerge from? What's the origin? Where did it come from? How did it get into the human population? So we got some early reports about where this thing came from. So based on looking at and comparing the genome of this SARS-CoV-2, this new virus, to others that are known, the first data that came back was that the closest match was to an existing bat coronavirus. And that really hasn't changed. So the belief is still that there is definitely a connection here of some sort to bats. And again, the closest match across the board is this bat coronavirus to SARS-CoV-2. So they're very close in sequence. So bats definitely play a role here. The closest match of this CoV-2 virus, not surprising, is SARS-CoV-1. Although there's quite a bit of difference between SARS-CoV-1 and CoV-2. And we'll talk a little bit more about that. But the closest match to a human virus is that SARS-CoV-1. Now, a new paper has come out here in the last week or so that talks a little bit more about the origin. One important thing about this paper is this, that its conclusion, and it's very clear that this virus was not designed. It doesn't make sense. What we're seeing in the genome of this virus does not lead anyone to believe that this is something that was designed in the lab. This is a virus that has emerged as a result of evolution in organisms, and it has now emerged and is circulating in the human population. This was not an intentionally designed, intentionally released virus by any means, or at least the data certainly does not indicate that it was intentional. This is a naturally occurring virus that has evolved and emerged. So really the conspiracy theories that this is somehow a biological weapon or some form of terrorism, there's just not data or evidence to support that. Again, this paper was published very recently, as all of these papers, but based on the analysis, there were some new things that came to light. So we had the data on the match with bats, but when you look at just the spike protein, just that particular protein that makes contact with the host cell, things are a little bit different and a little bit more interesting. So here's the data. Again, don't freak out. If you look at the top figure, all it's showing is a diagram of the genome of this particular virus, this SARS-CoV-2. And you notice the spike protein there is highlighted in red. Well, the figure at the bottom is just a zoomed in figure of just a few amino acids in that particular protein. Again, remember, proteins are made of amino acids. So what this figure at the bottom is showing us, the little boxes are around the 6 amino acids that specifically, the 6 amino acids in the spike protein that specifically make contact with the receptor on the host cell. So those, the amino acids in the boxes, and each amino acid, there are 20, as we know, each one has a unique 1-letter abbreviation. So this is identifying, with the box around them, the 6 amino acids from the spike protein that make contact with the receptor on the host cell. All right, so when you compare then this SARS-CoV-2 with the other known viruses, what you see is that only one of those 6 amino acids that are critical for making contact with the host cell receptor, only one is the same between SARS-CoV-2 and the closest relative, this bat coronavirus. So that kind of raises some questions of like, well, if these amino acids are so critical for the virus to bind to the host cell, only one of the 6 is the same? Interestingly as well, when you look at SARS-CoV-1, again, there's only one. So if you compare the little boxes, there's only one of the 6 that are the same. Again, kind of raises questions about, well, how really similar are these? And did COVID-2 come from this bat if, again, only one of these amino acids is the same? So the interesting thing that came out of this is that there is a 6-for-6 match with pangolin. And so pangolins are these particular scaly anteaters. And again, coronaviruses infect a wide variety of animals. And so what they found is that there's a 6-for-6 match. These 6 really important amino acids are exactly the same as the ones found in pangolins. So there's some new data to suggest that maybe, although it is believed that bats play a role and maybe they're the ultimate reservoir, but potentially the transmission more directly to humans were through these scaly anteaters, the pangolins. And I just saw a paper that came out this morning, and so I threw it in here. And it is a paper, again, coming out of China, where they have found SARS-CoV-2 related coronaviruses in these pangolins. Again, there's a picture of what they look like. They're scaly anteaters. And these particular guys that they used for the study, they were obtained in China. These particular pangolins are smuggled in. They are, at least from what I read and understand, they're relatively prized, both for their medicinal properties as well as potentially for food. And so these guys are desired, they're sold in the markets, they're smuggled in, and these scientists were able to take samples from some of these smuggled in pangolins, and they've identified related SARS-CoV-2 viruses. So there's more data emerging that potentially these guys, these scaly anteaters, as weird as that might be, these guys could be the route of transmission or the way that the virus emerged from the animals into humans. And so pangolins right now are those that are at least the data supports that those are the leading candidate at this point for the animal that ultimately transmits it potentially from a reservoir in the bat into humans. So new data that just came out this morning, again, identifying these SARS-CoV-2 related viruses in pangolins. Okay, so that's a bit about the origin, where they might have come from. Now let's kind of talk about the spread. So where is CoV-2 spreading? How is it spread? And again, you've read much on this, so some of this will be review, I hope. Others, hopefully, will be some new information. So here we go. You know, essentially, this stuff. So we talked last time about how viruses are shed from somebody who is infected, and then you pick it up from them. And so we do recognize that SARS-CoV-2 seems to be almost exclusively shed in respiratory droplets, so coming from nose and throat. I just saw a paper this morning where they tested the tears of people infected with SARS-CoV-2, and they could not find any virus. So it doesn't appear as though it's being shed in tears. So largely, again, respiratory droplets through coughing or sneezing, or so you can get infected directly. If you're close enough to somebody and they cough or somehow emitting respiratory droplets, you could, in fact, breathe those in direct inhalation. Again, that would require some pretty close contact. So very likely the more common way, although, again, there's no real data on this, but the more common way that you get infected is that somebody coughs on some surface or coughs into their hand and then touches a surface. And then, of course, you come along and you touch that same surface, and then you touch your mouth or your nose or your eyes or whatever, and that transmits the virus into you, and then the virus binds to the host cell receptor, and then the infection begins. All right. So if that is the way that this virus is transmitted, then how stable is it? So if somebody does cough into their hand and then touch a doorknob or touch some surface, how long does it stay around? So here's a paper that gives us some pretty decent ideas. So as a general rule, envelope viruses are not all that stable outside of the host. So that's a general rule. And coronaviruses are enveloped. They have that membrane around them. And so, again, they're not terribly stable outside of the host. So, okay, how not stable are they? Well, again, new data comes out. And so here's what we see, that essentially CoV-2 is as stable as CoV-1, at least in the same range. So what does that mean? Well, it means this. So when you aerosolize it, and this was done in a lab setting, keep in mind this is not coming out of somebody's mouth, but when fine particles aerosolized, they found that it stayed for about 3 hours. Again, I don't know how applicable that is to a normal environment, but that's what they found. So about 3 hours in the air. And this was like going to the very end of detecting any sort of existing virus. Certainly from the very beginning, the virus is deteriorating, but up to 3 hours, they could detect the presence of the virus still in that aerosol. Cardboard. So they found about 24 hours, the virus was able to survive on cardboard. So at 24 hours, they were still able to detect virus that would be viable for infection. Plastic and stainless steel, 2 to 3 days. So again, the virus is deteriorating as soon as it's getting on those surfaces, but after a few days, they were still able to detect virus that could cause infection. So hopefully this gives you some kind of idea of how long the virus can exist on surfaces, and therefore that kind of dictates how long between somebody depositing that virus onto a surface and you being able to pick it up. Okay, so we talked last time about reproduction number. So reproduction number, again, is this. It's the average number of people that would be infected from each infected person. So if I'm infected, what's the average number of people that I will transmit the virus to? And this is huge. It has lots of implications to predict spread, implications on the potential for spread. And so estimating this or getting an idea of this is pretty important. As I mentioned last time, measles is incredibly contagious and has a huge R0, a huge reproductive number. So something in the teens, like 18. So again, on average, one person with measles can transmit it to 18 people. Thus, the vaccine being pretty important. Influenza, typically seasonal flu, we're talking about less than 2, maybe 1.4, 1.6, something like that. So not a terribly high number. SARS-CoV-2 in this particular paper that was published, the estimate was about 2.6. As we're going to see in some modeling data, this number is really, really important. And it's not, again, a number that we are putting onto the virus. It's just a number that is dictated by the virus. But it has huge implications in terms of our ability to make decisions and make predictions about how it's going to spread, how many people will get infected, what kind of measures need to be taken. So we're going to come back to this. I'm going to show you some modeling data that uses different reproduction numbers. And you see very different results depending on how those reproduction number changes. So for SARS-CoV-2, at least an initial estimate, 2.6, that's the average number of people that will be infected for each person that gets infected with the virus. All right. So one of the questions is this, of course. The original SARS, we had 8,000 people worldwide, 800 deaths. So 8,000 people worldwide. Right now with SARS-CoV-2, confirmed cases, we are at about 500,000. Why the huge difference? Why is this virus spreading so much more than SARS-CoV-1? Well, the reality is that the data will come out eventually, and we just don't have a great idea of exactly what's happening. But there is at least some data that kind of will give us at least a little bit of an idea of where we are, what's happening, and maybe give us some insight as to why this is the case. So here's some data that indicates this. So this was a paper, again, published just on the 13th of March in Science by Rapp et al. And what they have found is that the spike protein of SARS-CoV-2, this new virus, binds to its receptor, ACE2, 10 times more strongly than the original SARS. So again, although these two viruses use the same receptor, ACE2, SARS-CoV-2 binds to its receptor 10 times more strongly than does the original SARS. So what does that mean? Well, we don't really know, but we can speculate or at least hypothesize that this could be a significant reason why the spread is more significant. This could imply that you need much fewer numbers of virus particles, fewer numbers, fewer virions in order to cause an infection with SARS-CoV-2 as opposed to CoV-1. So this could be, again, an indication of why we're seeing so much more spread with this particular virus than we did with CoV-1. Okay. This has been a big topic. So we recognize that there's probably a large percentage of people that are asymptomatic. So what's the role of asymptomatic people in the spread of this? So what I mean by asymptomatic is just that, somebody who is not symptomatic but has been infected. So they are infected with the virus, but they are not showing symptoms of the disease of COVID-19. So what's their role? Well, this particular paper published on March 16th gives us some idea. So this is data out of China, and what it's indicating is the level of stealth spread. And what they've indicated is that about 86% of cases in China, prior to the travel ban on January 23rd, 86% of cases were undocumented at that particular point. So 86% of cases were unconfirmed. So a significant number of folks that had been infected that were probably asymptomatic. Another thing to recognize is that those folks, though, were seemingly about 50% less contagious than those that were documented. So although they were asymptomatic, they were less contagious. And so that probably makes sense. But here's the big piece of data that's really important, that about 80% of documented infections were tied back to, or at least believed to be, from undocumented cases. So that is a huge number, obviously. So again, 80% of documented infections were caused by somebody who was undocumented, an unconfirmed case. And so this is kind of why they're referring to this as stealth spread. The spread of the virus from people that are essentially asymptomatic or undocumented to other people. So this is a significant issue and is a significant issue here in the US. The reality is that unless you're doing testing of everyone, you really have no idea of who has been infected and who hasn't. And even those that are showing symptoms aren't able to get tests. And so it's a real significant problem if a large percentage of new infections are coming from those that are unconfirmed or untested or even asymptomatic. It's really difficult to control the spread of something if you don't know who's been infected and who hasn't. So this is a real significant problem. And again, from this paper, data out of China indicates that a large percentage of people that are getting infected from folks that are unconfirmed, asymptomatic. All right, so this applies to you guys. There's been a lot of press on this as well. So, okay, fine, it spreads and potentially from asymptomatic people. Who gets it? Well, what I want you to focus on here from these numbers from the CDC, and again, they're now a couple weeks old, but look at the numbers in parentheses in this little table. You can see that out of 2,449 cases, and this was in the US between February 12th and March 16th, you can see that the age group with the most cases, the 20 to 44 age group, 705 cases compared to the other groups. So it really is important to recognize that it is a mistake to think that younger people are not getting infected and it's only older people. Younger people, again, are definitely getting infected with this virus. Sorry, I lost my slides there a second. Okay, I'm also having, okay, there we go. I wasn't seeing your questions. I apologize for that. So let me take a pause here and answer some of these questions. So will mortality rates likely fall when we count those who haven't been tested because they didn't show serious symptoms? So this is a really interesting question, Aaron, and there's been a lot of press on this as well, that, oh, once you include everyone, then the mortality rate will go down. Well, potentially in theory, but as this thing spreads, if our healthcare system gets overwhelmed, then we could see the mortality rate increase like it is in Italy. So I think it's kind of, it's a mistake to assume that mortality rates are going to fall from where they are now, because that's not been the case in Italy. And so we're just not sure. Is the R value place specific like the mortality rate? Yes, it can be. And Noah, to answer your question, sometimes we see the R naught change even as the spread continues, as the outbreak continues. That hopefully, as we practice social distancing and limit contact, we can drop that R naught value. So why would somebody be asymptomatic? Another question. Good. Somebody would be asymptomatic, again, if they're infected with the virus, but if the virus replication and infection of cells is just not causing enough damage to have some symptoms, or maybe your symptoms are relatively mild. These are questions that, again, we really don't know the answer to all that well. So why two people infected with the same virus, why someone would exhibit more symptoms than others? Could part of that have to do with behavior? Yeah, yes, potentially, Laura. So we do know that, probably smokers and people that are have particular behaviors that are damaging their lungs are probably going to be maybe more susceptible, but if they do get infected, more severe illness would be predicted in that case. Why is that age group so much broader than all of the others? That's a great question, Cameron. it would be great to have data that's broken down a little bit more, because I know you guys don't want to be associated with guys in their 40s. But it is important data to recognize. So are the numbers different from 20 to 30 and 30 to 40? Again, this is just the data that the CDC has collected and grouped, and so we don't know. But yes, it certainly could be quite different. And so I'm not sure why or what the reasoning is, but that's the reality. The point here I'm trying to make is that the 20 to 40 group is not immune from getting the virus. So yes, it is a broad range, but it's important to recognize that that group is a significant group that's getting infected. Can the virus that has died on a surface still cause harm or maybe provide immunity? So not really immunity in the sense. So for some coronaviruses, I believe the genome itself can be infectious, but for the most part, if the virus is not viable on that surface any longer, the probability that you're going to get infected is really, really low. Good questions. I apologize that the chat was not filling in for me, so I'm sorry I missed those questions. All right, so now that's a breakdown from the CDC of the groups that are getting infected. Let's look at severe cases. So with those that have been infected, how severe is the disease? So that takes us, Allegheny County numbers as of last night. I forgot, I just put this slide in. So here's where we are in terms of Allegheny County. And again, these numbers, I think, reflect that, again, it's not just the elderly that are getting infected. We are seeing numbers, again, 30 in that 25 to 50 age group. I realize it's a big age group, but again, it's important to recognize that there are a significant number of confirmed cases in younger people. And so it's not just the elderly. Again, we see, 15, 30, obviously a small sample size, but relevant to us. These are the numbers from Allegheny County. So now, what about severe disease? So the graph at the bottom, obviously, again, broken down by these same categories. This is from the same paper, the same data from the CDC. What I want you to see here is this. As you look at the light blue or gray, whatever color that is, you can see that's the number of hospitalizations in that particular age group. And then you can see ICU admissions in the darker color. And then finally, deaths in the darkest blue. And there's some trends here. And what I want you to recognize is that hospitalizations and ICU admissions are not necessarily trending or correlating with age. You're still seeing a significant number of hospitalizations and a significant number of ICU admissions in younger folks. It's not just those that are 70, 80 or above. Now, it is important to recognize that deaths, again, here in the US between February 12th and March 16th, deaths do seem to correlate with age. So the older you are, there is a correlation that there are more deaths in those older age groups. But when it comes to those requiring hospitalization, well, even backing up, those that are infected, those that require hospitalization, and those that require even ICU admission from that hospitalization, there is, again, a significant number in the younger age groups. So we are seeing severe disease among younger people. Again, this is data from the CDC. It's now a couple weeks old, but it at least shows us that, again, the younger age groups are not completely immune from, number one, getting infected, number two, having a severe, significant illness as a result of this particular virus. Okay. Now, if you do get exposed, how long should the quarantine be? In other words, what's the incubation period for this? So again, reviewing from last time, incubation period is that time from infection to the onset of symptoms. And so this particular paper that came out, and again, there's the link for it. And what it indicates is that the median incubation period for this particular disease, for COVID-19, is about 5 days. So that's the median incubation period. So that, again, that's the time from infection to onset of symptoms, about 5 days. Now, when you kind of expand that out, well, okay, fine. How long is it possible that this incubation period stretches out? their data indicated that about 98% of patients will develop symptoms within 11 and a half days. So even extending it out, about 98% of people are showing symptoms within 11 and a half days. So what this tells us is that kind of the current recommended quarantine of 14 days, if you believe you've been exposed or if you're in an area where somebody has, or where there is a significant number of people infected, the quarantine or the self-quarantine should be 14 days, and that should cover the incubation period. If you have been quarantined for 14 days, at that point, it's important to recognize here, though, at that point, yes, you were not infected by the person way back, assuming that you're not asymptomatic, but that doesn't mean that now you're immune and you can kind of, head out. So it's important to recognize that that 14 days is justified with data. The median incubation period is 5 days. So does incubation period vary with age? Is a question coming up in the chat. That I'm not aware of. I don't know if they had that data in the paper or not. That's a really good question. I don't know the answer to that, so I don't want to speak on that. So Timothy, why? What about the remaining 2%? I think they were a little bit longer than 11 and a half days, but again, without the paper in front of me, I'm not exactly sure. So how likely, another question, how likely is that you would be completely asymptomatic? It's a great question. We just don't know. Again, without more universal testing and without data coming from other countries that are doing more universal testing, we just don't know the proportion of people that are infected but remain asymptomatic. And for your additional question of, could you expect to see at least very mild symptoms? Potentially, but again, we just don't really know. So these are great questions. And again, we just don't know the answers to some of these. Eventually we'll know, or at least, and much of this could be corrected as well. I mean, that's how science works. All I'm showing you here is just the latest data, or at least stuff that's things that have been published. But again, that could change as more data rolls in, and that's how science works. We make decisions with the evidence that's available to us, but that certainly can change as we get more and more samples and more countries analyze, more people analyze, more data. The family I'm staying with was exposed about 10 days ago. So yeah, more than, again, I would say this, but again, it's not certain. I would say more than likely, if nobody in the family is showing any symptoms after 14 days, it's likely that you were not infected. Again, it's not absolutely certain. What we need is this. So the current tests, to be clear, the current testing is for the presence of the viral genome. So it's really testing for the presence of that viral RNA. So it's looking for the presence of the virus in the person with that nose-throat swab. What it is unable to determine is whether you were exposed at some different time. And again, if you've been exposed and you're asymptomatic, you're not currently being tested. What we need are antibody tests. We need serological tests. So we need blood to be drawn, and we need to look for antibodies against this particular virus. When we do that, that will then tell us how many people have been infected but didn't show symptoms. Because again, right now, we're only testing those that are showing symptoms and have likely been exposed and meet a whole series of criteria. So again, eventually we hope that we can get that serological data to get a better idea of actually how many people were infected, how many people were asymptomatic, and begin to put some of the pieces of the puzzle together. Just right now, keeping up with the people that are showing symptoms or need treatment, that's the number one priority. Trying to figure out who's been infected that's asymptomatic. That's just not something that we can focus on right now in the US because we were behind on the testing. And so now it's just a matter of trying to deal with the problem in front of us and worry about the other issues later. But hopefully we will eventually have that data. Those studies will be done. We just don't have that data right now. Great questions, guys. Great questions. All right. Let's look at control and prevention. So that's some things on the spread. Let's look at what is our ability to control this thing? What's our ability to prevent this thing? Well, going back to the last lecture, SARS-CoV-2 really is like the 800-pound gorilla. It sort of goes where it wants, it does what it wants, and we don't have much that we can do to stop it. So we have no current vaccine, we have no FDA-approved treatments, and it appears as though we don't really have any previous immunity to this. So again, we don't really know exactly, but it would appear potentially that infection with previous strains of coronavirus hasn't really helped us much. So this is a brand new virus that the human population has never seen before. And so again, it's kind of like the 800-pound gorilla. It's going and doing what it wants, and we have very little ability to do anything except to deal with the destruction that's left in the wake of the gorilla. And so really our only option is just to stay away, just avoid it. Try to not get infected. That's really all we can do at this point. But there's obviously been a ton of debate, lots of stuff out there on what should we be doing? How long should we be doing it for? Are these measures effective? Who even knows? Why are we closing down my favorite pizza place? Whatever. It's important that we kind of, again, base these decisions on data. So what do we have? Well, this is a paper that just came out, and I really think it's really interesting stuff. So it's modeling. So it's based on computer simulations that I won't pretend to understand, but the data I think that they got from this modeling, really, really interesting. So yes, Ethan, we're going to get to chloroquine and hydroxychloroquine here in a second. What this looked at, what they tried to do was they did some modeling, and this was specifically for Singapore. And the table that you can see in front of you looks at two different scenarios, and they actually did three. They did simulations with an R naught of 1.5, an R naught of 2, and an R naught of 2.5. You see baseline numbers indicating number of infections predicted if nothing is done. Then moving to the right, quarantine, that's the number of infections if you can quarantine those that you recognize have symptoms. You can then see the impact of quarantine plus school closure, quarantine plus workplace distancing, quarantine plus school plus workplace. So all of them combined, what are the projections or predictions? Again, this is all computer modeling. So what you can see here is that if you just kind of look at some of these, look at the top line, the total number of infections. With an R naught of 1.5, the modeling study indicates that if you do all of these things, you can cut that number from 279,000 predicted infections down to 1,800. And that 1,800 represents kind of an average. They did simulations over and over and over. I want to say like 1,000 simulations for each of these or something like that. So you can see very effective measures. R naught of 2.0, you can see that it's a little bit more challenging, because with the same measures in place, the baseline goes up from 279 to 727. Combined intervention would reduce it to 50,000. So what are some of the lessons here? Well, let's move on and look at some of their other data, which I think is really interesting. So their modeling data does indicate that these methods can be very, very effective at limiting infections. Here's an interesting thing. So this is a map of Singapore. On the left, and I apologize that you probably can't, the font's really small, but I wanted you to just see the figures. On the left column, that's the baseline. That's no intervention. That's what you would predict the spread to be if no measures are taken and everybody is free to go to school, go to work, no social distancing, do your thing. On the right is the result of the combined intervention. So quarantining those that are test positive, and workplace distancing and school closure. At the top row, those are R naught of 1.5. The middle row, R naught of 2.0. The bottom, R naught of 2.5. Again, the measures are all taking place comparing left to right, but you can see the impact of differences in R naught as we make predictions and model. So going back to the estimate that the R naught of this thing, the R naught of SARS-CoV-2 is 2.6, you can see that even with the measures in place, the spread is still going to be significant. Whereas if the R naught is 1.5, the measures are really, really important and can almost prevent or at least greatly reduce the number of infections. So it's important to recognize that R naught has a huge factor, and everything that we can do to try to limit the spread has a huge, huge impact. So let me now go to some additional data from this modeling paper that I think is even more interesting. Answering the question, or trying to answer the question of what is the impact of the percent of people that are asymptomatic. So the modeling that they did was with an R naught of 2.0, at least that's what I'm showing you here, an R naught of 2.0, and an asymptomatic population percent of 7.5. So 7.5% of the people were asymptomatic. So what I want to show you here is that if you look at the left column, that sort of jagged graph, the dark line at the top of that shows the level. So that would be the infections per day if nothing is done. And then the different colors that are filling in there, that's an overlay of all of their simulations. So if you just look at the very top one on the left, the dark blue is showing what happens if there's nothing, no measures taken. Then that red line that you see under it, that's if it's you institute quarantine for those that are diagnosed. Going down, school closure. That's what happens if you quarantine and you do school closure. Going down, workplace distancing. So quarantine plus workplace. And then all the way at the bottom, that's the difference. That's if you do all three. So again, the darker jagged line at the top, that's no intervention. And then the darker line under that in the middle of the shading, that's the end result. And that, so on the left, that is infections per day. On the right, that's total infections up to 80 days. So they did simulation for 80 days. And again, the dark line, that's what happens if you do nothing. And then the shaded part where it's darkest, that's going to be the most common simulation. So that's an overlay of all the simulations that they did. So where it's darkest, so either red or green or whatever, that shows you, and so essentially the difference between the darker line and the shaded part, that's showing you the impact on total infections as a result of these measures being taken. Now, so this is asymptomatic 7.5% with an R naught of 2. But let's kind of look at what would happen if that percent changes. Oh, let me answer a question first. So since just quarantine creates a massive difference, why are they doing all the other measures for only a small advantage? Or am I reading the data wrong? So it's not necessarily that you're reading the data wrong. This is, and they do go into this in a little bit more detail in the paper, that quarantine plus school closures plus workplace plus combined approach does have a pretty significant impact on the numbers. You're not reading it wrong necessarily, but let me show you what happens if the asymptomatic percent increases. So they did simulations all the way up to 50%. So I'm just showing you data for something in the middle. And I hope this begins to kind of get at your question a little bit more. At the top now, again, you see that the difference is much smaller between the total and those that were quarantined. Again, these are just projections. This is just computer modeling. This isn't real life, but this is just trying to model and see and predict how effective these measures would be. So in this case, the problem is that the greater the percent that are asymptomatic, the greater the numbers in terms of daily cases and total. And that's just because you can't quarantine people that you don't know are infected. And so this is the problem. The greater the number of people that are asymptomatic but still spreading the virus, the more difficult this thing is to control. If you can't identify who has it, you can't really stop it. And so you can see here that when quarantine isn't terribly effective because the asymptomatic number is high, then you can begin to see school closures, but especially workplace distancing really drops the numbers. And then if you do everything combined, you see it's pretty significant. So so you weren't reading the data wrong. And that was kind of a key, I think, for SARS-CoV-1, where it was perfectly evident the people that got it had these really high fevers, they had the cough, and so you put them in quarantine and you stop the spread. I think the challenge is now that we have a significant number, and again, that's backed up by the data from China, we have a significant number of people that are asymptomatic that don't believe they've been infected. The reality is they have been, and they are thus capable of spreading it. And this modeling shows us that, again, if that asymptomatic number is really significant, then just doing quarantine isn't enough. Again, if we don't know who's been infected, the best case scenario is for us just to not come in contact with each other. And this modeling supports that. It shows us that in those cases where the asymptomatic numbers are high or higher, then distancing really is very effective in preventing spread. So another question, does asymptomatic number mean that they are in the incubation period or that they have never developed symptoms and it just passes? So that's a good question. They certainly in the incubation period, they would be asymptomatic, but what we really mean here by asymptomatic is that they really never develop symptoms of the disease. Good question. All right, so let's get to, so that's a little bit on prevention and spread. So I thought that modeling data was kind of interesting. Because that's really all we can do at this point is try to make predictions, try to use the tools that we have. People that are much brighter than I am are able to do these kind of things and give us at least some data to base our decisions on. So let's kind of finish up with treatments. So where are we? Well, here is the current list of FDA-approved treatments for COVID-19. And so if you want to jot those down, it's important that you memorize this very extensive and comprehensive list. Obviously, there are none, and that's important to recognize. There are, to be clear, there are things that are FDA approved. So there are drugs that are FDA approved, but they are approved to treat other things. They have not been approved by the FDA to treat the COVID-19 disease caused by SARS-CoV-2. And it's really important to recognize that. So there are no current FDA approved treatments for COVID-19. This is where words become very important. It's really important to, again, be clear what you're communicating to people who need information and need answers. And so it's, to be perfectly clear, there are no current FDA-approved treatments for COVID-19. All right, so that settles that. Where are we heading? Well, the WHO just announced this solidarity trial. And so what they're doing is they're getting many countries involved. So right now there's 10 plus, and they're testing 3 particular drugs. And these drugs have already been approved for other uses. They just have not been approved for COVID-19. So remdesivir is a specific antiviral drug that was used at least, I believe, a bit for Ebola, but it is specifically antiviral, so it may have some impact here. Additionally, Kaletra, that's the name that it's sold under. It is actually a combination of 2 different anti-HIV drugs. And so there's some thought, okay, maybe that can be effective, so we're going to try that. And then what you've heard about, chloroquine or hydrochloroquine, a derivative, these are anti-malarial drugs. Again, these are used as not only preventative measures, but also to treat malaria. They also, though, happen to be immunosuppressive, and so they're used for people with lupus, rheumatoid arthritis, and this has become the real significant problem, is that folks with lupus that take this, and this is the only drug that they can use to treat their lupus, many of them, and this is anecdotal, but many of them are being told that the future availability of their drug is uncertain. And so that's the challenge. The FDA-approved drug for their particular disease potentially is not going to be available to them because of some of the misinformation that's been out there. So Emily's question, New York Post article that reported that New York hospitals are using vitamin C through IV to treat. How does that work and why? Wow. I have not seen that. I will try to look at that. Again, based on what I've told you this semester, there is no evidence that vitamin C really does anything. So I have no idea why they would be doing IV vitamin C unless there is some, I just don't know. So I don't necessarily want to comment whether they are or are not. But that's something I'll follow up on and try to get you an answer for next Tuesday. I have no idea why they would be doing that. So another question, does ibuprofen make it worse? That's a great question. There's been some recommendations out there that it potentially does, but again, without any sort of significant clinical trials, we don't know whether that's the case or not. So it'd be a little bit premature to say that. I have seen some things. I think there may have been some recommendations to not use ibuprofen in the treatment because of the possibility that makes it worse. But again, I'm not certain on that. Again, I'll try to get you a more definitive answer on that for Tuesday, but at least what I know right now, there's not any conclusive data that says that it does or doesn't. Good questions. Love them. So here's the controversy with hydroxychloroquine. So this paper came out and it was from a French group, and here's the data. So 16 patients that served as the control, 14 patients that got hydroxychloroquine, 6 that got hydroxychloroquine and the antibiotic azithromycin. If you've ever taken a Z-Pak for any sort of infection, that's azithromycin. So here is their data, and it certainly looks a bit promising. But it's important to recognize that there's, again, only, 30 patients here. I think 4 dropped down or something like that. So we're talking about a very, very small sample size. Again, if you compare the black line, the control line, and so they're looking at the percentage of patients with, and so PCR positive, that's just an indication of a positive presence of the virus. So they've detected the virus in patients. So starting at 100% on day 0 and going to day 6. So what they find is that the control patients, 6 days later, 90% of them still have detectable virus. With hydroxychloroquine alone, they saw down to about 50%. And with the combination of hydroxychloroquine and azithromycin, they saw all the way down to nothing by day 5. So certainly promising data, but far from a nicely controlled, blinded clinical trial. And so to tell people that this is a treatment option is really, really reckless. This is, again, far from any kind of definitive data. It shows some promise, but that's all. It only shows us that there's potentially some promise, and this absolutely requires some further study. Now, a paper that just came out from China, again, all I could read was the translated abstract, but they did 15 and 15, and what they saw was the following. So they treated 15 people with hydroxychloroquine, they had 15 controls, and they saw the following. So in the time it took for patients to test negative for the virus, they found no difference. In the time that it took for body temperature return to normal, no difference. In the time that it took to see progress by lung CT scans, no difference. So again, the data from the French group is promising. The data from the Chinese group is contradictory. Again, we just don't know. So it is certainly warranted, further study is warranted, but it's really reckless, I think, to believe or to widely prescribe this particular drug. There's just not enough data, and there are some valid side effects. So really good question from Cameron. How would chloroquine do if malaria is not a virus? Excellent question. So malaria is caused by a single-celled protozoan parasite called Plasmodium falciparum or falciparum. So this particular protozoan, this particular parasite, causes malaria. Obviously, it's a living cell. Viruses are not. What it's believed to maybe do, and this is just maybe, it's believed that it perhaps lowers the pH of some of the small compartments in the cell that are required for viral replication. So that's a really general, generic answer, but it's believed to potentially have an impact to interrupt that replication cycle that we talked about in the last lecture. But again, that's largely speculative. We just don't really know. Number one, we don't even know if it works. And so number two, we certainly don't know the exact mechanism if it does. Great, great questions. So in terms of hydroxychloroquine, chloroquine, yes, very, very small data that suggests that it might work. But again, we just don't have enough data to know. All right. I just threw this in here to make sure you were aware there is a vaccine trial for COVID-19 for SARS-CoV-2. It just started in Seattle. They're recruiting 45 healthy people. They're going to break them up into 3 groups. They're going to give each group 2 different, or they're going to give each group 2 doses of the vaccine, and they're going to use 3 different concentrations, 3 different levels, or 3 different doses, looking for an immune response. So ultimately, at the end of this, they're looking for an immune response across those 3 groups. And not surprisingly, the goal here is to get an immune response, get antibodies made against the spike protein. So that's the protein that projects out from the virus. The antibody would bind to it and prevent infection or spread. So that vaccine trial has just started at Kaiser Permanente in Seattle. They're going to follow up with these patients for quite a while and see if they elicit some kind of immune response. Okay, so here's some questions that everybody wants to know. And we'll finish with these last few slides. How long is this going to continue? Well, here's the latest data. And this is from Hopkins with their coronavirus map and their data. So these are the top 3 countries, and we're right now 3rd. So China is at the top, Italy in the middle, and USA at the bottom. And so what these data, these are the combined number of cases. So these are the total number of cases. And the axis on the bottom, the dates are the same, so you can kind of line up and follow the progress. So you see in China, this outbreak, and this data goes from like January 21st. So this is basically like 2 months worth of data. So you can see in China that after about a month or so, the number of cases hit their peak, and then it kind of, the combined cases plateaued. So that's our hope. Our hope right now is that this thing will plateau at some point, but the question is when. Because when you look at Italy, which is in the middle, they're still certainly going up. And then, man, if you look at the US at the bottom, I mean, we are on a trajectory that is not slowing down, at least, well, yeah, we don't know. That's the reality. We just don't know. We know our trajectory is not good. The number of cases each day is really, it's increasing rapidly. And so there are some really significant questions there of when is this going to stop in the US? When are we going to see it level off? We know right now it's increasing at a really rapid rate. Let's look, so that was total cases. Let's look at increases per day. Again, same thing, China at the top, Italy in the middle, USA at the bottom. So we see the number of, so the increase in cases per day in China went up and then leveled off, and then you see that it's kind of been level or nothing for at least relatively recently. You see Italy has gone up, and then maybe, we just don't know, but maybe it's potentially beginning to tail off or plateau. That would be a really good thing. But here in the US, just yesterday, we had a record number of daily cases. So again, we are not, we're not out of this thing. We're not even close. We are on the upswing. So in terms of number of cases per day, we're increasing at the moment. So we just, it's important that we kind of stay buckled in and keep the measures in place to, at some point, again, China is our hope that this thing will pass. We will be able to take the lid off of some of the distancing and the quarantines. But right now, the data is certainly not suggesting that we do any of that. Let me finish with this slide just to try to get in, and then I will try to answer some of those questions. So how bad will this get? That's kind of the question you want to know. The reality is nobody knows. There's some predictions out there of when this thing will peak. And I've seen 3 weeks, I've seen 6 weeks, I've seen 8 weeks. Just in an article I read this morning, nobody knows. It's all speculative at this point. We just don't know. But what we do know is if this trend increases, really within the next couple of days, the US will be #1 in terms of total cases worldwide. We've moved up to #3, and we're right behind Italy and China. Again, you can see that China's cases have leveled off. And so the US definitely is on a trajectory to surpass those countries in number of confirmed cases. So again, we're dealing with the 800-pound gorilla here. So all we can really do, it depends on you, it depends on me, it depends on us. Think of any sort of, we use the term colloquially of something going viral. Well, think of a video or a post that goes viral. That requires individuals to continue to share and spread that thing. And so if you, and all you can do is just control your own interactions. And so the less that you share this thing, the less it will spread, or at least hopefully we can lessen the spread. And so I think that's kind of the punchline in all of this, is that a virus requires a host in order to replicate and spread. And so kind of the goal here is don't be a host if you can possibly prevent it. I know that it's a difficult time. I know this is challenging. It's challenging for all of us. But again, the data, finally, since we've gotten some testing going, we're now seeing the reality and seeing the numbers. And so until we can get a handle on this thing, the numbers are increasing rapidly. And again, yesterday was the largest number of cases per day, the largest increase in cases that we've seen in the US yet. All right, so let me answer a couple of these questions. So from Emily, given that there are so many unknowns with treatment, duration, and spread, what are your thoughts on how long Americans should stay home away from work, given the recent fall in the stock market and the new stat that 3.2 million have requested unemployment benefits? Do you think this could have a detrimental effect on the economy? So this is one, Emily, where I will stay in my lane, so to speak. The reality is, I just talked to my financial advisor yesterday, and he essentially said that we just don't know. Certainly, the economy is taking a big hit. We just don't know what the eventual impact would be. At this point, we just don't know how long we're going to be away from work, how long schools are going to be closed. We just don't really have those answers at the moment. And so the eventual impact on the economy is something I can't predict, and I'm not sure many people can. Another question, is the plateau due to their quarantine or some other cause we don't know? So yeah, if I assume you're talking about China, the restrictions that they put in were significant. Yes, they put in some restrictions and some quarantines that were very, very dramatic. And so, yes, I think we at least can attribute the plateau to that. They're just now starting to talk about slowly lifting the lid off and getting things maybe back to some semblance of normal. So it'll be really interesting to watch China, and as they do that, to see if cases come back up or not. That's something really interesting to keep an eye on. So James, could the extreme trajectory in the US be due to the wide availability of testing? Absolutely. So James, here's the challenge, is that because the testing was not available and people weren't tested early on, and because testing is limited, and because there are people exhibiting symptoms that can't get tested, and please understand, I'm not making a political statement here by any means. I'm just stating facts in terms of availability of testing. It wasn't there. And so we are sort of getting up to speed on testing as maybe the virus is spreading more. We just don't know. That's the problem, is that we don't know if the significant daily increases are due to active virus spreading, or whether they're due more to just now people getting tested. Again, we just don't know. And so we're not going to know that until testing is much more widespread and much more available. At that point, then we can get a better handle on it. Right now, we just don't know. All we can do is look at the data and say, wow, we're seeing these dramatic increases day by day by day. What that's due to, we don't necessarily know. New spread or just new testing, a combination of both, no doubt, but we just don't know how much to contribute to each one. Naomi, why is the U.S. spread so much worse than China when we have less people and less population density? Yeah, these are some great questions. And again, we just don't necessarily know. Now, again, China put in some very significant and almost immediate lockdowns, quarantines. We've been, well, I don't want to express an opinion. We've kind of scattered those out. They've been up to individual states. They've been up to individual towns, cities. So we have a lot of different people making decisions for their particular area. And so that's certainly a contributing factor. We've seen quite a bit of evidence of people still congregating, still going out, still going to vacation spots. And that's a significant problem because, again, even though we've just seen, even though they might be feeling fine, they very well could be spreading the virus to lots of people. So there's a real problem with that. At least here in Pennsylvania, the governor has been pretty clear. He really doesn't want to get to the step of having to enforce these things, but he will if people aren't complying. So the hope now is that we can all kind of do our part and comply so that there won't be a need for more kind of heavy-handed government action. But again, looking at the modeling out of Singapore, these measures do work. And yes, they're uncomfortable. Yes, it's detrimental to the economy. But again, we're talking about a significant number of lives and people that are affected. So from a health standpoint, these things are absolutely needed to try to protect those that are most vulnerable. And certainly, as Christians, I hope that we can recognize that is a very clear calling for us to protect those that are most vulnerable in these situations. But we'll keep an eye on this. We'll see what happens in China as they begin to lift some of these restrictions. But for now, here in the U.S., the numbers are certainly not indicating that this is leveling off or even slowing down anytime soon. So my prayer and hope is certainly that will be the case sooner than later. But the reality is we just don't know. All right, guys, I've gone over by a few minutes, and thank you for your questions. Please, what I will do on Tuesday, well, I'm going to stop the recording now, and then I'll talk a little bit about what we're going to do on Tuesday.